Hello and welcome back everyone to the conclusion of Chapter 3. So this has been a bit of a lengthy chapter, so let's just go ahead and finish off with a brief discussion on cell cycle control and stem cells in the body. So let's go ahead and rehash something that we have mentioned before, and something that you will, if you recall from your introductory biology class, you should remember quite well. So most of the roughly 30 trillion cells in the body are what we call diploid, meaning that they contain 2 copies each of the 23 unique chromosomes. So obviously that gives us a total of 46. So the question is, why do we have 2 copies of each chromosome, and where did we get them from? Well, that's a question that has a fairly simple answer that we have addressed before. In the process of fertilization, you get 23 chromosomes from your dad's sperm, and you get 23 chromosomes from your mom's egg. The 2 cells join and produce a diploid zygote, from which you mature and differentiate into a mature human being. So pre-existing cells, whether it's a somatic cell in our body or a germ cell that is going to participate in reproduction, pre-existing cells in the body can give rise to new cells by one of 2 different mechanisms. So most commonly, we're going to talk about mitosis. So the idea here is that both resulting daughter cells are going to be genetically identical to the progenitor cell, and they will each be diploid. So this is going to be a perfect mechanism for growth and repair. This is what we want to do things like generating new blood cells, repairing injuries to tissue like broken bones and cuts and scrapes and things of that nature. And then there's also going to be meiosis, which this has only one very specific purpose, and this is for sexual reproduction. So the only cells that are going to be produced via meiosis are going to be sperm and egg, the processes of spermatogenesis and oogenesis, which could be covered in a completely different discussion. So the difference between mitosis and meiosis is that in meiosis, each gamete, whether it's sperm or egg, should, statistically speaking, be genetically unique due to homologous recombination and a number of other different mechanisms that you probably talked about in general biology. And then the other difference is that these are going to be haploid cells, meaning that they only contain 23 total chromosomes, meaning only 1 copy of each chromosome. So the whole cell cycle can usually be represented as kind of a timer mechanism, and that's not a bad way to kind of look at it. So most of our focus, and we're not really focusing on the whole process that much anyway, but any focus that we do have is mostly going to be on mitosis, which is part of this larger process that we call the cell cycle. So somatic cells in your body, meaning cells other than the cells that produce gametes, which is most of your cells, somatic cells in your body, most of the cells in your body are going to fall into one of 2 different categories. Either they're going to be cells that are actively dividing, in which case those cells are going to be at some point in the cell cycle here, either somewhere in interphase, which is G1S or G2, or in M phase, which is mitosis. And then actually most of those somatic cells in your body are actually done dividing. They're what we call quiescent. There was a time in your development in which they were dividing, but they basically outgrew the need for that, so now they are quiescent and they never ever divide. So have you ever kind of heard of maybe cells in your body that you're kind of stuck with, you're never going to make any more of those cells? Generally, the tissues that you hear about about the most are going to be your brain and your heart. So however many brain cells you have right now, you're never going to have more than that because those cells cannot divide. The same goes with heart cells. So this is the reason why injuries such as strokes and heart attacks are so debilitating, because they cause the death of a lot of those cells, and those dead cells cannot be replaced because the healthy cells around them are not capable of dividing in order to replace the cells that were lost. Most of us dividing cells' time is spent in that interphase process. So mitosis is actually a very small little sliver of the cell cycle. So any given dividing cell in your body is somewhere in interphase, statistically speaking. So interphase is all about growing and making new materials like DNA, making new proteins, making new organelles, a lot of those anabolic reactions that we talked about in Chapter 1, and a lot of the dehydration synthesis reactions that we talked about in Chapter 2. What mitosis in phase is all about is taking those duplicated materials and using specific mechanisms that we're not really going to touch on here to divvy up those materials, divvy up the chromosomes, divvy up the organelles, and then using cytokinesis to make sure that we make two daughter cells that roughly have an equal amount of stuff from the original duplicated progenitor cell. So we're not going to talk about the whole process of mitosis or really even the whole process of cell cycle. So we're not going to talk about things like prophase and metaphase and anaphase and telophase and any of that. We're not going to talk about kinetochores or spindles or microtubules or anything like that. That's more of a biology topic. So we want to kind of focus on what is going to be specifically relevant to human physiology and pathophysiology. So we want our focus here to be on cell cycle control. How do we walk a fine balance in keeping yet another thing constant? In terms of homeostasis, another thing that we can keep homeostatic and keep constant is the number of cells in the body. We don't want to have far too many cells, but we definitely don't want to have not enough. So we need to have a balance between making new cells and balancing that with the death of old cells. So very classically, and you're probably aware of this, an imbalance in this kind of balance that we try to walk here leads to things like tumors and cancer, in which case cell cycle progression gets out of control and we make far too many cells than we mean to, and that's how we get cancer. So the way we usually deal with this is, as we said, we walk a fine balance. We balance the cell cycle by determining when is it appropriate to go through the cell cycle, and in other cases, we suppress the cell cycle to keep it from happening when we don't mean it to. So we balance this through what are called go and stop signals. A go signal would occur in a time in which we do want the cell cycle to occur. All other times, we have these stop signals up that prevent the cell cycle from going ahead. So any change in these signals can disrupt the balance and potentially lead to cancer. So in this way, we have two different types of cell cycle genes that make cell cycle proteins. Go signals are called proto-oncogenes. So when these are working the way that they're supposed to, we call them proto-oncogenes. When a proto-oncogene accumulates a bad mutation that causes it to be way too active, it causes it to promote the cell cycle when it's not supposed to, we stop calling it a proto-oncogene and we instead just call it an oncogene. Proto-oncogenes are good. Mutated oncogenes are very bad. They lead to things like cancer. On the other hand, stop signals are called tumor-suppressor genes. The way we can get cancer from these is a little bit different. If you get a mutation in a tumor suppressor gene, like p53, which is a protein that's mutated in a lot of cancers, a mutation in a tumor suppressor gene would cause a cancer if it removes its normal function. So if a tumor suppressor gene's stop activity suddenly stops working, then that can shift the balance towards cell cycle progression, and that could cause cancer as well. So your takeaway from this is basically we can get cancer in one of two different ways. Either proto-oncogenes become way too active and become oncogenes, or tumor suppressor genes lose their activity and we can get cancer in either of those ways. But before you go worrying too much, it is true that it's a very bad thing when we get bad mutations in these genes, and that can cause the cell cycle to get out of control and cancer to accumulate, but usually one mutation is not enough. Generally, a person is going to have to accumulate quite a few of these different mutations before they really start to accumulate cancer. There are a lot of kind of fail-safe mechanisms to make sure that one mutation is not going to be the end-all be-all. So the last thing that we want to talk about here is the process of differentiation. So this is going to lead us into a discussion on stem cells. So we've covered this before. When you were conceived by mom and dad, you at the end of fertilization existed as a diploid zygote. So you had a total of 46 chromosomes, 23 from the sperm and 23 from the oocyte, the egg. So the question that we have addressed before, and we'll go ahead and give ourselves an answer again, how did you go from a single-celled zygote into the full-grown, roughly 30 trillion cell adult that you are today? Well, we owe this to two different mechanisms. Many rounds of interphase and mitosis, which allowed us to go from 1 cell to 2 cell to 4 to 8 and so on and so forth. But also we rely on differentiation because, as we said, mitosis is always going to produce 2 cells that are genetically identical to one another. We could not have brain cells and heart cells and skeletal muscle cells and liver cells and things of that nature if every cell was identical to one another. So the process of differentiation allows cells to modify their internal structure so that they can take on a very specialized function, as we have discussed before. So a lot of differentiation is talking about stem cells. So you might have heard of stem cells. Stem cells were kind of a very hot topic in the news 10 to 15 to 20 years ago, and they still are very important. So let's go ahead and talk a little bit about different types of stem cells. So basically you can think of a stem cell as a very diverse type of cell that is capable of giving rise to other types of cells in the body. A stem cell, another way of looking at it, is a cell that is capable of differentiating. So we classify stem cells on the basis of how much potential do they have. So how diverse are they in terms of which types of cells they become. The word potential that I used is appropriate because we actually use that word to classify these cells. The highest level of stem cell is called a totipotent or pluripotent stem cell. Basically, these have the potential to differentiate into any type of cell in the human body. Now, this sounds like a really great deal, but here's the bad news. An adult human being does not have any totipotent or pluripotent stem cells. These only exist very early on in development, meaning right after the time that fertilization occurs. After a while, after several rounds of division and differentiation, these stem cells cease to be, and you become much more limited in terms of what stem cells can do. Now, multipotent stem cells do exist in some forms in the human body. These are stem cells that are capable of differentiating into a few different cell types, but not a whole lot. They're not capable of differentiating into every type of cell, but they are capable of differentiating into a few different types. And then most stem cells in the body are what we call unipotent in terms of they are already determined in what they are going to become. So they are fully differentiated, and when they undergo mitosis, they can only produce one very specific type of cell. One very important process that we will talk about several times throughout the semester, so this is a good time to go ahead and introduce the topic, is the process of hematopoiesis. So that word is kind of a tongueful, right? So hematopoietic stem cells are multipotent. And what the whole process of hematopoiesis is all about is generating the diverse different types of blood cells, red blood cells, white blood cells, and platelets. So hematopoietic stem cells exist in a few different places, but mainly in the red bone marrow, as we will discuss later on in the semester. Hematopoietic stem cells are multipotent in such that they can differentiate into all the different types of blood cells, like red blood cells, white blood cells, and platelets, but they cannot differentiate into things like brain cells or heart cells or liver cells. So when we're talking about, when we're talking about stem cells, we're talking about things called lineages. So a lineage basically gives you a roadmap that tells you what different types of pathways the differentiation can take to give you different types of cells. So the progenitor of the hematopoietic stem cell is called a hemocytoblast. You can follow a number of different pathways to determine which types of cells can be provided. One pathway, called the myeloid pathway, will give rise to red blood cells, which are called erythrocytes, and then most of the white blood cells, and then also some platelets, which are called thrombocytes. The lymphoid pathway will give you the other types of white blood cells. Mainly these are going to be your lymphocytes and a lot of your very specialized white blood cells like natural killer T cells. So this is just a little bit of a taste of stem cell physiology and a lot of cell cycle control. Again, we didn't want to really get into the nitty-gritty specifics of the actual mitosis and cell cycle process. We just wanted to give ourselves enough of an appreciation to understand some things moving forward. So this represents the end of Chapter 3. Chapter 4, which is coming up next, is going to be a rather short chapter, just giving us, now that we have a little bit of a foundation in cell biology, we can start looking at specific tissues. And then after Chapter 4, it should be clear sailing into looking at a number of different organs and organ systems, which is probably what your expectation was coming into human physiology class. So I will go ahead and sign off for now, and I look forward to seeing you next time. Bye-bye.